In this video, I'd like to talk about cell membranes and the structure of cell membranes and the relationships of the different lipid structures. So cell membranes consist of a bilayer, and this bilayer is defined by the tail-to-tail opposing phospholipids that form in the bilayer. Now, what is a phospholipid? A phospholipid is very similar in structure to a triazyl glycerol that we saw previously. It has a glycerol backbone, 3-carbon backbone. Two of the carbons have esterified fatty acids to them. And the number 1 carbon has a phosphate group with a head group. The head group, along with the phosphate combination, can provide charge and bonding in an aqueous environment, and so the head group is representing the head of the structure in the normal drawing. The long hydrocarbon tails of the fatty acids are represented by these tails in the membrane. So that means the intramembrane space is very hydrophobic because of the long hydrocarbon tails. The head groups on the outside of the cell, as well as the inside of the cell, can interact with salts and water through our normal 4 non-covalent weak interactions and remain soluble. So now, we also have in the membrane proteins, and proteins can be in different forms. They can be what are called transmembrane. Transmembrane proteins have structure that is exposed on the outside of the cell as well as the inside. The transmembrane portion of the protein tends to be an alpha helical structure, and you can predict that the R groups are going to be hydrophobic amino acids in order to remain properly bonded inside the hydrophobic tails of the phospholipids. We also have proteins that can be peripherally associated. These are called peripheral membrane proteins, and these proteins, you can imagine, will interact here at the interface with the polar head groups of the phospholipids. Some proteins can be embedded into the membrane. These are also termed peripheral proteins, but these proteins have a unique character that they can interact in the hydrophobic domain of the intramembrane space as well as phospholipid head groups that are polar on the outside of the bilayer. Now, these particular phospholipids and proteins have a capacity for lateral diffusion. Lateral diffusion in membrane is very, very rapid. Occurs on the time of seconds and less. Proteins can move also laterally in the membrane. But transferring a phospholipid, essentially flipping it from one side of the membrane to the other, takes a very long time unless we have the presence of enzymes called flipases that can flip essentially a phospholipid from one side of the bilayer to another. The enzyme flipases are required because of the polar nature of the head group has to be shielded from the hydrophobic domain of the intramembrane space in order for this transition to occur. Once you have flipases present, these transitions can occur very, very rapidly. Now, going back to our discussion about fatty acid structure, we talked about saturated fatty acids, and these fatty acids all have single bonds in their carbons. We talked about unsaturated fatty acids, And these are variable amounts of double bonds. Now, these two types of fatty acids work together in a biological membrane. For example, let's take a look at this membrane. Let's draw on this side here. We will draw the representation of a saturated fatty acid. Saturated means we have all single bonds. The hydrocarbon tail is long and straight. Now, if we have another saturated fatty acid here next to it, we see that these are packed very tightly, and those weak hydrophobic forces that are enabled bonding energy here can form numerous bonds between these two. Increase bond means increase strength. So under a situation like this, this decreases fluidity of the membrane, simply because we have an increase in the number of potential interactions and bonds in this hydrophobic domain. So an increase in saturation of our fatty acid hydrocarbon tails will lead to a decrease in fluidity in the membrane. Now, in order to increase the fluidity, we have to introduce something that will disrupt all of these well-formed hydrophobic bonds. This is where our unsaturated bonds come in. If we have a bond, double bond, here in one of our fatty acid tails, you'll notice that the distance between these two hydrocarbon tails is too great to form effective productive interactions. They could potentially form interactions here as the two hydrocarbon tails approach each other, but this region up here is too weak. That will increase fluidity because you've decreased the amount of bond energy. So decreasing saturation, which is also saying increasing unsaturation, will increase fluidity. The other molecular structure that we're going to introduce in here is cholesterol. Cholesterol has a 4-ring structure. One of the carbon positions on the ring has a hydroxyl group on it. The other end has a short little hydrocarbon tail. The cholesterol molecules, the size of it is enough to fit through 1/2 of the bilayer. If we take cholesterol and introduce it into the membrane, this is going to modulate fluidity. Now, what does modulate mean? If we have long saturated hydrocarbons next to each other, the cholesterol molecule will essentially disrupt some of that bonding to weaken it. If the cholesterol molecule fits its way into an area where we have long saturated hydrocarbons adjacent to an unsaturated, this can fit nicely into this spot and increase the number of bonds. So in certain circumstances, the cholesterol will decrease bond energy. Other cases, it will increase bond energy, essentially modulate. Now, how does this look like if we draw a graph of membrane fluidity that we're measuring versus temperature? Now, we know that temperature will act as the energy source to disrupt bonds. And so if we have a pure substance, pure saturated fatty acid, think back on organic chemistry. When you had a pure substance and you took its melting temperature, you had a very, very sharp transition between the solid and liquid state. That told you that you essentially had one molecular species in the tube. Same thing with saturated phospholipids. If all of our phospholipids are saturated, we will go from a solid to a liquid in a very short time, very short window of temperature. This would indicate a pure substance, just like in organic chemistry, is a full saturated phospholipid. Now the problem with this is that if our membranes were constructed of all saturated phospholipids, we would have a very narrow temperature range where our membranes would be a proper biological fluidity. We don't want them solids because we will not have lateral diffusion of proteins and phospholipids that are necessary for cell function. We don't want them liquid because there's a chance that the membrane could rupture under hydrostatic pressure and be too fluid. We want somewhere in the middle. But life is out here in this range. This is where our temperatures exist. So if we get too cold, our membranes could go solid or they could become too liquid. So just like in organic chemistry, when we introduce a contaminant of sorts into our pure substance, we increase the temperature range of the phase transition from solid to liquid. So now this is saturated. So now let's do saturated plus unsaturated. What we will see here is essentially a little bit more of the width of the temperature range for phase transitions. So now we have a phase transition that has increased the width. So in this range here, with the introduction of an unsaturated fatty acid, we have essentially increased the temperature range for our optimal membrane fluidity. Now we can take this one step further and add cholesterol. Cholesterol serves to modulate the fluidity, as we discussed, by fitting in between the phospholipids and either disrupting interactions or enhancing interactions. The end result of this is going to be further increasing the temperature of the phase transition. So by increasing and having saturated, unsaturated fatty acids as well as cholesterol, we have a sufficient amount of molecular interactions here to cause a perfect membrane fluidity case among the temperature range that we live at. We don't have anything too solid at low temperatures or too liquid at high temperatures.